Dear participants, we will start the session within 5 minutes. Let everybody join. I am audible to everyone.
Yes.
So now we are starting the session. Okay. Very good evening to all respected dignitaries, resource person, faculty member, researcher, student, and dear participants. It is my great pleasure to welcome you all to today's session of 30-day summer training program under International Krishi Manthan 2026. We are delighted to have you all with us as we continue this enriching journey of learning, knowledge sharing, and professional development. Today's session is graced by an esteemed academician and researcher, Dr. Vikas Sharma, who is serving as an assistant professor at Swami Keswanandan Rajasthan Agriculture University, Bikaner. Dr. Sharma research expertise lies in microbiology and plant microbe interaction, which are crucial for sustainable agriculture and improving crop productivity. He has made significant research contribution in areas such as plant growth, promoting rhizobacteria, including Bacillus, Pennybacillus species for sustainable agriculture, seed priming using cyanobacteria, plant microbe interaction for nutrient availability and biofortification, crop health and disease study, environmental issues like ecosystem service loss due to paddy straw burning. His research publications have received a strong academic recognition with over 600 citations and a growing research impact reflected through his ANS index and I-10 index. Some of his highly cited work include research on PGPR for sustainable agriculture, ecosystem impact of crop residue burning, and microbial approach to improve crop performance. Dr. Sharma work clearly reflect his dedication to a sustainable agriculture, environmental protection, and microbial innovation in crop improvement. We are truly honored to have such a dynamic research and academician with us today. I am confident that his insight will greatly benefit to all participants. Without taking much time, I respectfully invite Dr. Vikas Sharma to deliver his expert talk. Sir, the stage is yours. Yes, you can please continue, sir.
Thank you, sir. So, first of all, I welcome all the participants and all who have joined, including host and all the researchers. Myself, Dr. Vikas Sharma, working as assistant professor at Swami Keswanand Rajasthan Agricultural University, Bikaner. And I am working in this university for last 14 years, and my post graduation and PhD is from ICR, Indian Agricultural Research Institute. So where I did my lot of research, and here I am also working on this, including PGPR and biocontrol agents, and also involved in teaching and all other activities. So am I audible to all? I think I am audible.
Yes, sir. Pretty much audible, sir.
Yes. So this is, I think, diverse group, but all are means belongs to, I think, agriculture, or some others are also there.
From agriculture, biotech, the
Okay.
STEM science.
Okay. So shall I share that presentation screen? That sharing option is not active. So I request host Mr. Rathore Mehta that please, my option of sharing, it should be started. Presently, that screen sharing option is not working. Please switch on your microphone. You are not audible.
We have started the function, sir. You can.
Okay, but this function. No, again that sharing option. This one.
Yes.
Okay. Thank you. Entire screen or only window.
Select this and ask.
So I think that screen I have shared, it is visible to all the
Yes, sir. It is clearly visible. Sir.
Yes. So more of the interaction it will be there, and some discussion. I'm working on plant growth promoting rhizobacteria or microorganisms. So I will try to link or share my experience, or whatever that sort of knowledge it is prevailing in the literature. So today's topic of discussion is plant growth and health promoting microorganisms for sustainable agriculture. So before coming to that PGPR, I would like to some discussion about soil, because that most of other microorganisms are also there, but in PGPR, that rhizobacteria, they are living in rhizosphere region. So for the agricultural purpose, that soil, it is very much fundamental, or it is, as we know, that it is very much complex system and also a dynamic biological system. Why it is complex and dynamic? Because it is not an inert material. It is dynamic because of a lot of living organisms are residing. It is soil, it is a house, or in these are inhabitants are there in soil. Due to them, it is a dynamic system, and it is very much complex system because thousands of thousand species of macro and microorganisms are residing in the soil. So for taking one example, that soil, that is upper layer of the earth, and even only 1 gram of the soil, it contain more than billion of microorganisms. So you can say that whole human population in one spoon of soil. So human that the population is equivalent to the 1 spoon of soil. So this much of microorganisms are residing in the soil. So this is so the microbial activity and microorganisms are most beneficial for crop, and only 1% of Earth's microflora that have been identified and characterized so far. Why it is this? Because most of the microorganisms, those are not cultivable. We don't know that how to cultivate in laboratory or artificial conditions. So the process is in the progress that various microorganisms, we are just characterizing that which kind of media, which kind of nutrients, which kind of conditions are favorable to cultivate these microorganisms. So only 1% of the this diversity we have been able to explore. 99% of the diversity of soil microorganisms or these plant growth promoting microorganisms is yet to study. So, in talking about India, it is the one of the 12 countries of having mega diversity centers. So, because of that, due to various climatic and conditions, agroclimatic conditions, various soil types, or due to that, it is mega diversity center. India is a very big mega diversity center. And interaction among plant, soil, and microorganisms, it is very much needed to interpret it, and up to now, or it is not interpreted well because of lack of resources, and that this is recent science. So a lot of work is going on. We will discuss in coming slides that what kind of work is going on, and what are the benefit these are causing to crop, and what benefits are provided to soil. So what tripartite kind of interaction among these three partners. So only few of them plant growth promoters that inhabiting the rhizosphere. Now these are characterized, and India's microbial diversity has not been adequately worked on till date. About the microbial diversity, we are what kind of diversity are there in soil, and what functions these are doing. We will discuss that these are three category, or majorly four category of microorganisms or organisms in soil are there. That macrofauna, size that is more than 2 millimeter, that those are visible with naked eyes, like moles and slugs or termites, ants, earthworms. These comes under macrofauna. Then little smaller, that mesofauna category, less than 2 millimeter, then but more than 0.1 millimeter. These comes mites and some other organisms, which comes under category microfauna and macrofauna. These are mesofauna. Then most important, which are there, microfauna and microflora. So microfauna, these are size less than 0.1 millimeter, like nematodes and protozoans, but these are animal-like structures. So these are microscopic. Similarly, microscopic organisms are microflora, which is most important, that bacteria, fungi, and viruses. Since their size is less than 0.1 millimeter, so these microorganisms, we need microscopy to study these, to see them. Uh, our human eyes are not capable to observe these with naked eyes. So microbial diversity, that how much diverse these microorganisms these are, qualitative or quantitative diversity is there in the soil. So they play a key role in soil structure as well as function in natural as well as man-made ecosystem like agricultural ecosystem where cultivated soils are there. Natural ecosystem means that forest soils or pasture soils where no intervention, human intervention or minimum human intervention is there. But managed agriculture soil like our fields or cultivated fields where crop or orchards which are have been raised, there also microorganisms play a very important role. So what are their role? They maintain biogeochemical cycling of nutrients, various nutrients like nitrogen, phosphorus, sulfur, carbon. These nutrients are they're cycling in the soil or in the ecosystem. It is being done by microorganisms. They degrade organic compounds or naturally occurring polyphenolic compounds which are being added to soils or naturally occurring in the soil. Why? Because when we harvest the crops, their roots or some other organic matters are added into the soil, or these macro and microfauna or flora which are residing in soil, upon their death they are also mixed into the soil. So they also become a part of soil organic matter. When these are being degraded, they are also become nutrients for the other microorganism as well as plants. So this is very much important, and 80 to 90% of the processes in soil are mediated by richness of microorganisms. That is the level of their function or level of their importance in the soil, that up to 80 to 90% of various functions which are being done in soil due to their richness of microbes. How much microbes are important to the soil. So when we discuss microbes and plant, their interaction, then that word it comes plant microbiome. You might have heard about human microbiome or gut microbiome, that in human gut various kinds of microorganisms are residing, and those are functioning, those are performing various activities like some vitamins are being provided, or some conversion, fermentation or other conversions are being made. Nutrients are being provided to human through these microorganisms. Those are part of gut microbiome. Similarly, in plants, plant microbiome is also there. Each plant have their unique or characteristics microbiome. So this microbiome decide that how resistant that plant will be there, how its growth it will be there, how much sturdy that plant it will be there, or in abiotic or biotic stress if it is there, then how that plant is going to fight with all these kind of stresses. So how much vigorous its growth it will be. So microorganism, they colonize different plant parts. These microorganisms may be fungi or bacteria, some primitive kind of bacteria like archaeobacteria. So all these microorganisms, it may be actinomycetes, it may be cyanobacteria. These microorganisms which are in close proximity with plant, these are known collectively they form plant microbiome. That is, I think it will be clear to all that what are plant microbiome. Before going to next slide, we will just discuss that microbiome that emerged as fundamental plant traits for determining beneficial or some negative effect with plant growth or health or productivity point of views. And what this microbiome we can divide into various parts depending upon the plant parts, above ground or below ground plant parts are there, or whether these microorganisms are residing on the surface of plant or within the tissue of the plant. So some are epiphytic. So these epiphytic microorganism on the above ground plant parts like leaves, like stem, like flower, like fruit, on these parts are these microorganisms are residing. So it may be phyllosphere on leaves, okay, or on stem. Airborne it will be there, microbiome also will be there. Then endophytic microorganism, these reside within the plant tissue, within the plant parts, various plant parts on within the roots, it may be within the leaf tissue also. So these are endophytic microorganisms. Then one of the important aspect is rhizospheric microorganisms. It means rhizo means roots. So microorganisms which are in close proximity with plant roots. and those are direct under influence of plant roots. Those are known as rhizospheric microorganisms. So depending upon their position with plant interaction, epiphytic, endophytic, and rhizospheric microorganisms are there. Similarly, the biomass and composition of this microbiome means that how much biomass is there, and composition means how what is the diversity, different kind of bacteria, different kind of fungi or protozoa. These microorganisms are residing various kind of micro, means it is qualitative and quantitative. Microbiome, they largely affect the interaction between plant and environment. These are microorganisms, but they decide interaction of plant with their immediate environment, that how that plant it will respond to environment, that is being decided by these microorganisms, whether it may be positive way, it may be in negative way. So it is important to discuss this point. What is plant microbe interaction first, before deciding that how these are influencing plants growth. What is plant microbe interaction? So this picture is clearly showing that what kind of various kind of interactions are there with plant and microorganisms, that below soil what kind of interactions are being done. So first we can say that plant microbe interaction, it plays a important role in effective root colonization as well as biocontrol. Biocontrol, it is needed that because all the microorganisms are not beneficial for plant. Many are the pathogenic, many are harmful. So control of these harmful microorganisms is essential. Then rhizosphere is populated with countless organisms, numerous microorganisms are there. And already we have discussed that merely 1 gram of soil, it contains billions of microorganisms. So these are most populated ecosystem. Then rhizospheric community, it competes for space as well as nutrient with indigenous microorganisms. Indigenous microorganisms are apart from that plant, whether plant is growing or not, these microorganisms are always living in the this soil. These are known as indigenous microbes. But what are the rhizospheric community? When plant is growing, its roots existing in soil. So the area which is influenced by directly under influence of plant roots, that is known as rhizosphere. And what kind of microorganisms are residing in rhizosphere? Known as rhizospheric community. So this rhizospheric community is entirely different from the bulk soil. That is apart from the plant, means distant apart from the plant roots. Then this plant provide an auxiliary environment for microorganisms in return, because that microorganisms also benefiting plant, then plant also providing shelter and the competent environment to these microorganisms, or we can say rhizospheric community, and also enables beneficial microbes survive and proliferate along the growing of the plant roots. So in this diagram, we can see that various kinds of activity. In A, we can here we can see that plants exude phenolic compounds, various phenolic compounds to discourage the germination or growth of the plants. So it is known as allelopathy. It one kind of plant microbe interaction. Another kind of interaction you can see that root feeding larvae, various kind of larvae, that stimulate the production of some volatile compounds, volatile organic compounds, VOCs, that attract entomopathogenic nematodes. So this kind of negative kind of interaction is there. Then root exudates. What is root exudate? We haven't discussed yet. Root exudates are the liquid which is secreted by the plant roots into the soil. So that is beneficial for microorganisms. That is that plays a food for microorganisms. And it is said that around 2 to 10% of the photosynthesis which are being made by plant, which are secreted in the soil in the form of these root exudates. So these root exudates promote colonization of rhizobacteria and initiate the nodulation of rhizobia. So this is helpful. Then another kind of interaction in root exudates, they induce hatching of cyst nematodes and attract juvenile nematodes towards the roots. So this is also a negative kind, but this is signal for these nematodes which are not beneficial for plants health point of view. Then root cap is the site of most active exudation. We can see here that among the whole root, that cap area of the root, that is most harbor most of the population of microorganisms. Then in another interaction, we can see that strigolactone, some of the chemicals are exudated through plant roots. They induce hyphal branching in arbuscular mycorrhizal fungi, AM fungi. These AM fungi are the fungus which are helping plant to uptake various nutrients, because plant roots are they have limited surface area. They cannot reach to deeper layer of the soil, but these mycorrhiza, AM fungi or mycorrhiza kind of fungus, they make some symbiotic interaction with plant roots, and they are benefiting plants to provide the nutrients. Then another one interaction, you can see that same strigolactone in the root exudates, the host exudated by host that trigger germination of some parasitic plants. Parasitic plants like striga, total root parasite, that can be also induced through these strigolactones. Then what are plant growth promoting microorganisms, PGPM? Because everyone is talking about PGPM. So these plant growth promoting rhizobacteria or plant growth promoting microorganisms are those microorganisms that colonize rhizosphere or rhizoplane region. Rhizosphere means soil with plant root, that area which is immediate vicinity of plant roots. But rhizoplane is the area, it is known as surface of plant root. It do not include soil, only surface of the plant root. What kind of rhizobacteria are there? It may be Acinetobacter, Agrobacterium, or Azotobacter, Azospirillum, Burkholderia, Rhizobium, Thiobacillus, Pseudomonas, Bacillus, various kinds of rhizobacteria are there. Then biocontrol fungi are also plant growth promoting microorganisms. Though these are not directly involved in the plant growth promotion, but indirectly they are hindering growth of the pathogens like trichoderma, various fusarium, aspergillus, arbuscular mycorrhiza, penicillium, or some actinomycetes are also there, like Frankia, streptomyces. Those are not, these are some examples of species. Those are plant growth promoting microorganisms. What are their function? Exert beneficial effect on plant growth. Positively, they influence. They compete over the existing rhizosphere communities which are not beneficial. Then influenced by plant type, plant age, and soil type or agricultural management regime. Plant type means some are leguminous, some are gramineous plants, some are some other species. So plant to plant, and their population vary. Kind means qualitative and quantitative. Plant age also means in nursery or younger plant, these harbor different kind of community. But when plant gets older, then community totally changed. Soil type and agricultural practices, cultivation practices like tilling or all these practices also influence their community. What are their mode of action? How they benefiting plants? So plant growth promoting microorganisms, there are three ways. They provide nutrients to plant or protect plant from biotic stress, and another way is protect plant from abiotic stress. So you know very well that biotic stress are caused by living organisms and abiotic stress are caused by non-living causes like temperature, frost, or drought. These conditions are causing abiotic stress. So directly that providing nutrients to plant that in that area, we know that various symbiotic and non-symbiotic nitrogen fixing microorganisms are there. Symbiotic, it may be rhizobium or bradyrhizobium, various species, but non-symbiotic like azospirillum, azotobacter, or acinetobacter, these are species are there. They are involved in fixing atmospheric nitrogen and providing to the plant, which is assimilated by plant in ammoniacal form or nitrate form. Then production of phytohormones in forms of indole acetic acid or gibberellic acid, or reducing the amount of ethylene. Then solubilization of phosphates and other minerals like zinc, potassium. These are insoluble form of phosphate, potassium, zinc. These are not available to plant, although these are present in soil. But when it is being solubilized by these plant growth promoting microorganisms, these are readily available to plant, and plant growth, it will be benefited in positive way. Then another way is protection of plant from biotic stress. Biotic stress means various pathogens or insects, those are causing harm to plant. So in this way, they are producing siderophores, some proteins which are quenching iron. So you know that already soil is lack of iron. So when iron is being quenched by these microorganisms, that iron deficiency, it will be there, and harmful microorganisms will not be able to survive in the soils. Similarly, cyanides producing, antibiotics producing, then some enzymes like glucanase, chitinase, these enzymes, they inhibit fungus or various pathogens by degrading their cell wall. Then induced systemic resistance, ISR, or systemic acquired resistance. These are various kinds of resistance are being provided by these microorganisms or plant growth promoting microorganisms. So these directly or indirectly influencing plant growth. Similarly, in abiotic stress, that altering the levels of some various enzymes like stress enzymes like peroxidases, superoxide dismutase, proline. These are accumulated in plant on when in sufficient amount. Then effect of abiotic stress like drought, like high temperature, like salinity. These effects are minimized by these microorganisms. Production of acc-deaminase, polyphenol oxidase. So acc-deaminase also a kind of, and this is one of the enzyme which are reducing the amount of ethylene produced by plant. Why? When that ethylene is produced by plant during the time of senescence, but immediate precursor of the ethylene is ACC. So this acc-deaminase degrade this ACC, then ethylene production, it will be less and plant will be healthy. Then another way is biological nitrogen fixation. So already we have discussed that the two kind of nitrogen fixation is there, symbiotic and non-symbiotic. In symbiotic rhizobium or various species of rhizobium like bradyrhizobium, azorhizobium, mesorhizobium, and other than rhizobium, frankia is also symbiotic nitrogen fixing bacteria. Some are free living because that symbiotic like rhizobium, they are forming nodules and living with roots. But very many are the free living like Azospirillum, Enterobacter, Klebsiella, or cyanobacteria. Various photosynthetic microorganisms are also there. Those are fixing atmospheric nitrogen parenchyma. So overall, it is estimated that biological nitrogen fixation, it contribute to 65% of nitrogen it is being used by agricultural soils. So this much of importance is there. If these were not there in our system, then what was the pressure? What imagine that what kind of pressure it would have been on the nitrogenous fertilizer like urea and other all these. So these are very helpful microorganisms, and it is estimated that around more than 3,000 crores of rupees per season are being saved by the activity of this biological nitrogen fixation. So this is helping that farmers and government because there are a lot of subsidies being provided by government also. These are some mechanisms of biotic stress management. We have already discussed that biotic stress is being caused by various pathogens or insect pests. So various mechanism when the pathogen is attacking on plant, or whether on above ground or below ground parts, some pathogen associated proteins are activated, and these are exciting some intrinsic systemic resistance. This causes accumulation of phenolic compounds or some response like jasmonic acid or ethylene. These kind of compounds are being produced by plant in response to biotic stress. So in this way, these microorganisms are going to help plant through excitation, various catalase or superoxide dismutase enzyme, or it quenching reactive oxygen species or peroxide, various means deleterious chemicals which are being produced due to abiotic or biotic stress, that is are being managed by these microorganisms. Similarly, abiotic stress is also caused by it may be drought or salinity, or various causes are there, high or very low temperature, frost injury. So these microorganisms means plant growth promoting microorganisms or rhizobacteria PGPR, they produce various hormones like cytokinin. So cytokinin, it is reducing the effect of abscisic acid. So in case of drought or various reactive oxygen species are being produced in case of drought. So these microorganisms are able to manage a situation of drought, or various antioxidants are produced by these microorganisms. So these antioxidants are able to quench the reactive oxygen species rose. So similarly, ACC-deaminase, we already discussed that amount of ethylene being reduced, or some volatile organic compounds, they manage that high affinity potassium transporters. So in this way, abiotic and biotic stress is being managed. Some beneficial effect of microorganisms, plant growth promoting microorganisms, are production of indole acetic acid. So this indole acetic acid, it is involved in promotion of root growth or rapid re-establishment of lateral growth, main root as well as lateral roots, fast growth of young roots, and causing ability to anchor with themselves with the soil. So this indole acetic acid being produced by plant growth promoting microorganism. Also some amount of gibberellins, some microorganisms, PGPM, are able to produce gibberellic acid or gibberellins, various kinds of gibberellins, and it initiate mobilization of storage material in seed during the germination process, because at the time of germination, stored material in form of starch or various form protein, that is, it should be mobilized, so that a lot of energy is required at the time of germination, because no leaf is there, no photosynthetic activity is there. So stored material, it helps that germinating seed to make vigorous plant. It causes elongation of stem and pollen tube growth at the time of pollination. Then similarly, some PGPM produce cytokinase, which involved in cell division or shoot formation or delay senescence of the tissue. So that vegetative part is maintained for longer time. So more photosynthesis it will be there, and more production it will be there. Then phosphate solubilization is also various phosphate solubilizing microorganisms are there. Various bacteria like Pseudomonas fluorescens or some fungi also, Aspergillus species or some species of fungus, are able to solubilize insoluble phosphate in the soil, so that a plant can uptake this solubilized phosphorus. They improve phosphorus availability to plant, and the photosynthesis and crop yield or seed formation, it is enhanced. Some microorganisms are able to produce siderophores. We already discussed that these are iron quenching molecules produced by PGPM, which contribute to disease suppression, role in plant growth stimulation, also iron uptake by plant, because that whatever amount is that has been coinced by these siderophores, that is used by microorganism, as well as it is provided to the host plant also. Ancient production, various microorganisms produce cyanide kind of compounds. They suppress fungal disease or plant pathogens on the root area, and also inhibit plant pathogens. Then hydrolytic enzymes, they hydrolyze fungal cell wall. These enzymes include glucanase, lipase, amylase, pectinase, cellulase, protease. These enzymes are involved in the polysaccharides. Next, many are producing exopolysaccharide (EPS), and they form biofilm. So that anchorage with plant roots is enhanced by PGPM. They hold free phosphorus or insoluble in the soil. They shed desiccation, stress, defense, and response to during the infection process. Various vitamins are produced, like B group of vitamin. It may be vitamin B12, B5, B1. So these enhance resistance to drought and oxidative stress. Oxidative stress, it may be caused by various biotic and abiotic stresses. Organic acids produced by PGPM, various organic acid like lactic acid, oxalic acid, gluconic acid, these are being produced in inorganic phosphate solubilization, and also involved in enhanced plant growth. Antibiotic producing microorganism PGPM, majorly some fungus and actinomycetes are there. They inhibit soil-borne pathogen or bacteria. What kind of antibiotics they produce? Bacillomycin, entruin, phenagine, or mycos, subutylin, agrocin. These kinds of examples of antibiotics produced by plant growth promoting microorganisms. Then various antifungal compounds are also being produced by PGPM. They inhibit growth of fungal plant pathogens like glucan synthesis inhibitors. Then volatile VOCs, volatile organic compounds, are also produced by PGPM. without directly coming into contact with plant, these volatile compounds they fetch the plant and trigger growth promotion to the plant or enhance intrinsic systemic resistance. These involves ethylene, butanediol, acetone, butane, 2-butanol, like these are volatile compounds. So this importance, that what amount of the PGPM are there and what is their importance to plant growth. So when we are studying these microorganisms, PGPM, so how these soil microbes are being characterized, how to characterize this. So starting with polyphagic approaches for identification of these plant growth promoting microorganisms. First, we take soil sample. Two methods are there: culture dependent method and another one is culture independent method. Culture dependent method means those microorganisms which are already known to us and we know that how to cultivate them in laboratory condition, artificial condition. So we go for directly culture dependent method because this is easier one. It involves cell extraction, then further serial dilution. That serial dilution is we are further going for plating on nutrient agar or like other potato dextrose agar, Kenites media. Then after plating, that bacterial or fungal isolates are there. We isolate them or purify them and identify the species based upon some probe design or some in situ hybridization or these. Then culture independent method are being used for all those microorganisms which are very difficult to cultivate in the laboratory condition. So directly from soil samples, DNA or RNA extraction is being done. Then PCR or RT-PCR in case of RNA. And two strategies may be there: directly cloning, sequencing, or probe design. Or some community fingerprinting methods are like DDG, OTGG, or RISA, RFLP. Then further amplification, sequencing, and probe design, or similarly through in situ hybridization, these are being identified. So in culture dependent methods, we already have studied that in conventional method of microbial identification, this is culture dependent because this is conventionally we are using in our laboratories. First one is biotyping, like biochemical growth requirement, that how much amount of sodium chloride it needs, amino acid, how much various kinds, how many kinds of sugars these are fermenting, hydrolysis of compound like starch or various compounds, gelatin. Then environmental condition, that how much pH it needs it's for optimum growth, how much temperature, what is the range of temperature, is it resistant to any antibiotic or susceptibility to any bacteriocins produced by other organisms. Then second one is serotyping. Serotyping, it includes examination of cell surface antigens, and like ELISA, enzyme amino sorbent assay, RIA, agglutination test, or fluorescent or enzyme labeling. These are serotyping. It involves identification of based upon the protein. Then phage typing. Phage are the viruses or bacteriophage which causing death of which are means eating that bacteria. So characterized by pattern of resistance or susceptibility to standard bacteriophage or viruses which already have identified. This phage typing is done. Another method, it may be community level physiological profiling, that is also known as biolog, multitop, matrix assisted laser desorption, ionization, and time of flight. Multitop is also being used for identification of new microorganism. Then another one is FAM, fatty acid methyl ester analysis. This is used for community study. All these are used for community. Then culture independent method, where culturing is not possible. We don't know that how to culture these organisms in laboratory condition. So metagenomic study is being done directly from soil sample. DNA or RNA is isolated, and PCR, we go for a PCR and RT-PCR. And microbial community fingerprinting, various techniques are available as we discussed: DGG or TRFP, ARDRA, RISA, SSCP, that is single strand conformational polymorphism, and microarrays as well. These are methods. Then what are the methods for enhancing beneficial microorganisms? Okay, how to study them, what is their importance? Then in soil, what are the actual methods to enhance their population? Because these are beneficial, so their population, it should be higher in the soil so that plant can be benefited. So crop soil health management, there are two kinds of biological health management practices. One is black box approach, another one is silver bullet. So black box, it is natural kind of method, and silver bullet, it is augmentation. Artificially, we are increasing the population. So in natural management of resident population means that a native population or indigenous population that of microorganisms, these are being managed. In silver bullet, introduction of specific microorganisms to reduce the disease, like biocontrol agents are introduced on the seed surface or in the soil, or likewise in with irrigation water, any way. While transplanting, we can use these microorganisms. So that is one term is that suppressive soil. Which is suppressive soil? Soil in which that disease development is suppressed, even though the pathogen is introduced in the presence of a susceptible host. What are the three conditions for a pathogen to cause disease? That susceptible host, virulent pathogen, and favorable environment. All these conditions are there. Even though suppressive soils, they are the healthy soils which reduce the incidence of disease. Then another second one that we discussed, the first method, black box approach, like managing resident microbes. It is biological. What are these bacteria, plant growth promoting rhizobacteria, or fungi, or actinomycetes? So these are suppressiveness of soil-borne plant pathogen. What mechanism it is there? It is competition of native microflora, or these PGPR, with pathogens. It may be amensalism, antagonism, parasitism, intrinsic systemic resistance, or systematically acquired resistance. Some cultural methods means we do not directly introduce microorganisms. Already present microorganisms are those are made to flourish in the soil, like cropping effect, crop rotation, and cover crops like green manures or clean planting material. While starting that seed or planting material, it should be disease free. Then make site unfavorable for disease, like mulching and stacking. These are methods. These make the site unfavorable for pathogens. Then some soil amendments are like application of compost, vermicompost, organic mulches, neem oil, neem cake, FYM. All these methods comes under black box approach. Then silver bullet approach, these are two kinds of approaches are there: single PGPR and cocktail of PGPR. So what is single PGPR method for disease management? These are some examples, like in rice, some blast, sheath blight, sheath rot, stem rot. Those it is reported by various authors, like Pseudomonas fluorescens, Pseudomonas putida, Bacillus species, various species of Bacillus or Enterobacter. These PGPR species are able to manage these diseases of rice. Similarly, in sorghum, PNP, chickpea, pea, soybean, various species of Pseudomonas fluorescens, various species of this Bacillus, and even Azotobacter or Azospirillum. These are nitrogen-fixing microorganisms. These are also able to manage root rot disease in pea. Similarly, various cash crops like coconut, groundnut, potato, these are various PGPM are reported to control various kind of diseases, including root rot or crown rot or damping of in nursery, or some wilt kind of disease. These diseases are being controlled by these managed by these microorganisms. Then silver bullet, second one is silver bullet approach for plant growth and yield. It is various reports are there that various kinds of rhizobacteria like Bacillus subtilis in reported in wheat that enhance seed germination. Similarly, Azotobacter in mung bean and pea, they enhance growth. And a Bacillus cereus in soybean enhance nodulation and root. Then Azospirillum, Azotobacter, or Pseudomonas in sunflower enhance the growth and high seed yield. Similarly, Pseudomonas fluorescens or phosphobacteria in ginger and black pepper or various turmeric, these spices are there. So enhanced nutrient mobilization due to these Pseudomonas fluorescens and phosphobacteria, especially phosphorus, and that is resulted in higher yields in terms of the production. Then if someone want to work that how to isolate or characterize these microorganisms, some briefly, very brief, we will discuss this isolation and identification of these. So isolation, it is done by serialization on nutrient agar or other media like candidates broth media, and identification according to the standard Burgess manual of systematic biology. Then study of plant growth promoting potential of bacteria. After isolation, how to study that these are then some mechanisms are there, some tests are there, some protocols are there for siderophore production, indolacetic acid production, or phosphate solubilization. So these are standard method. Then hydrolytic enzyme production properties. So various enzymes like cellulase, amylase, protease, lipase are there, which if those are present in these microorganisms, so it may be these may be potent for biocontrol as a biocontrol agents or antagonistic potential of bacteria against various diseases like macropomena, rhizocotonia, coledotrichum, sclerotium. It has been reported, and various methods are available. Then characterization. After identification, that characterization of PGPR. So various you can see that in first picture you can see antagonistic activity by rhizospheric bacteria by dual culture method, that one pathogen is there and at where that PGPR is there. So how it is controlling. Then siderophore production for iron chelation. Then phosphate solubilization, that how much phosphate, insoluble phosphate, whether it is able to solubilize this insoluble phosphate. Then production of enzymes like indolacetic acid. These are the pictures you can see. Characterization. One example we can see that like trichoderma. So it is biocontrol agent. How we characterize what are the characters able to survive under diverse environment condition, harsh condition, even high temperature or drought condition, alter microflora composition of the roots because pathogens are suppressed by this trichoderma. These enhance nutrient uptake or solubilization, enhance root development, root hair into deeper root formation, induce systemic resistance, enhance tolerance of crop to biotic and abiotic stress. And another is mycoparasitism, means produce some enzymes like chitinase that attack on fungal cell wall and its skeleton, insect skeleton, because it is also made of chitin, insect skeleton as well as fungal cell wall. It is made of chitin. So chitinase enzyme is produced by this trichoderma. Then other beneficial effect, tolerance towards biotic and abiotic stress. It is producing chitinase, then reduction of ethylene, production of volatile organic compounds and oxygens. And here you can see that in control that root growth is limited, but in trichoderma inoculated plants, that profuse root is there. Some are limitation of individual biocontrol. Two studies we discussed, that individual PGPR, one is cocktail or consortium. So what are limitation of individual single microorganism we are using? There is low tolerance or stability under diverse soil environment, low competence to rhizosphere community, native community. Then these exerts low or short duration defense, low level of antioxidant activities, and also shorter shelf life of biocontrol agents in general, or storage duration, improper storage is there, so its shelf life is also less. But limitation with microbial consortium, when we are using more than one microorganism, so it may be when we are using 3 or 4 microorganism or cocktail, then there may be incompatibility among those microorganisms, or certain microbial consortia were unable to comparable effects, or lack of quality product, because it is now new avenue of research in case of microbial consortia. So why these microbial consortia needed? Conventionally single culture use, but now various cocktail of microorganisms is being used. What is this microbial cocktail or consortium? It is group of compatible microbe. First thing, foremost thing is these microorganisms or group, it should be compatible with each other for plant growth promotion, nutrient mineralization, and antagonistic effect against pathogen. They are making healthy soil for sustainable management of crops. So priming of these microorganism or cocktail, it may enhance soil suppressiveness or strengthen the plant against various biotic as well as abiotic stress conditions. So these are some examples that Pseudomonas, Bacillus, Brevibacillus, that Burkholderia, all these various institutes are making these cocktails or consortium. These are some researchable issue, industrial issue, or regulatory issues are there. So not our. Finally, I want to conclude this as that next generation of sustainable agriculture. Plant probiotics, you have seen that Yakult or various products in dairy, you can see that probiotic lassi, charge, and various probiotics are there. So these now there is time to use these plant probiotics in form of plant growth promoting microorganism, biofertilizer, like using microbes under soil to help the plant. Then emphasis must be given to the development of cocktails of potential bioinoculants having multi-trait properties in the existing IPM technologies, integrated pest management technology. Because multi-trait, when we are using someone, it will be involved in nitrogen fixation, someone in potassium solubilization, phosphate mobilization, or zinc, someone it will be biocontrol agent. So multifunction, it will be beneficial for plant. Then combination of both traditional as well as modern scientific approach for soil and crop health management, it can be taken or carry forward for our agricultural land and more productive and sustainable way in times to come or in the near future. So in this way, I conclude my seminar. So I want to thank each and everyone for your patience or here that listening to it is very carefully. So thank you, thank you.
Thank you very much, sir, for your insightful and enriching presentation. Your research-based perspective on plant microbe interaction and sustainable agriculture have provided us with valuable knowledge and practical understanding. Now, we would like to open the floor for interaction. Dear participants, if you have any question, please free to unmute your mic or type your query in the chat box. Sir will be happy to address your questions.
Yes, yes, please.
I have given the access of mic. You, dear participant, you can unmute your mic and ask your query or write in the chat box.
Yeah. Also, since that discussion should be there, healthy discussion, it is beneficial for me also, for all the participants. So any kind of query, please don't hesitate. I want to thank all the host and that EcoFast Agri Solution for giving me to deliver this lecture and giving me opportunity to deliver this lecture. I'm very
Yes. Dear participants, have you any question you can freely ask with our esteemed sir.
Yes, any kind of that basic question or application related question you may ask, no problem. Manisha Sharma, she has written, what is the role of actinomycetes in plant growth? Okay, Manisha. So I want to say that actinomycetes are kind of bacteria, actinobacteria. Nowadays we are calling these actinobacteria. So these are a specific kind of bacteria, similar, many of, like in illness or some plant, Frankia. Frankia is one of the species which is acting as plant growth promoting actinomycetes. So it is helping that rhizobium, it is forming a symbiotic relationship with legume crops. Similarly, some actinobacteria or actinomycetes like Frankia, it is also making symbiotic relationship with various non-legume plants like Alnus or Adler. So these are helpful in nitrogen fixation. Then some of the actinomycetes like streptomyces. These are producing antibiotics, various antibiotics. So these antibiotics produced in the by these actinomycetes, which are very helpful for control of these pathogens. So indirectly, if pathogen number is less, disease incidence it will be less. So plant growth, it is helping plant growth and higher yield in the crop. Okay, so I think that Manisha, you are satisfied. Okay, okay, thank you, thank you.
Any other query, dear participants?
सर एक क्वेश्चन था।
बिल्कुल पूछिए।
सर अगर हमने मतलब किसी प्लांट के अंदर हार्मोन की कमी है तो हमने वो हार्मोन का इंजेक्शन लगा दिया, लेकिन फिर भी अगर वो मतलब कमी उसकी फुलफिल नहीं हुई तो मतलब क्या कारण हो सकता है कि वो फुलफिल नहीं हो रही प्लांट तब भी? जैसे कोई मतलब फ्रूट है वो डार्क रह गया तो हमने उसके अंदर हार्मोन डाल दिया, इंजेक्शन लगा दिया, लेकिन उसके बाद भी मतलब वो नहीं बढ़ रहा, तो क्या दिक्कत हो सकती है?
उसमें मैं भी एक तो देखो कारण ये हो सकता है कि एक्चुअली वो पर्टिकुलर जो डोज है ना, कि जिस काम के लिए आप दे रहे हैं वो डोज दूसरा, उसको स्टोर करने के लिए खास करके जो हार्मोन्स है, ग्रोथ हार्मोन्स है, तो उनके अंदर क्या होता है कि अगर स्टोर प्रॉपरली नहीं किया हुआ तो उनकी इफेक्टिविटी बहुत कम हो जाती है। और जनरली ये क्या है, इंजेक्शन के फॉर्म में नहीं देते हैं और ये स्प्रे किया जाता है बहुत ही डाइल्यूट फॉर्म में, है ना, पीपीएम या पीपीबी कंसंट्रेशन में, पार्ट्स पर मिलियन या पार्ट्स पर बिलियन, मतलब हार्डली 1 लीटर अगर सॉल्यूशन बना रहे हैं तो मिलीग्राम्स में इसकी रिक्वायरमेंट होती है। तो वो एक मेथड ऑफ एप्लीकेशन में भी हो सकता है। टाइमली अप्लाई नहीं किया, वो डे टाइम में अप्लाई कर दिया, बहुत ज्यादा हाई टेम्परेचर है, हार्श कंडीशन है, तो यूवी रेडिएशन के अंदर ये डिग्रेड भी हो जाते हैं, तो वो भी एक रीजन हो सकता है। कंपेटेबिलिटी का भी हो सकता है। तो ये कुछ रीजंस है, कुछ प्रिकॉशंस रखने पड़ते हैं, है ना, और मोस्टली स्प्रे के फॉर्म में ही देना चाहिए।
सर अगर ओवरडोज हो जाए तो क्या करना चाहिए हमें?
ओवरडोज हो जाए तो उस केस में तो एक्चुअली उसका वैसे तो डायरेक्टली कुछ ऐसा नहीं है वाटरिंग और इस तरह के, अदरवाइज क्या होता है कि ओवरडोज होगी तो उसके कुछ ना कुछ हार्मफुल इफेक्ट तो उसमें दिखेंगे, बिकॉज होता है ना कई बार कि हाँ, लेकिन कुछ है पर्टिकुलर हार्मोन के कुछ एंटीडोट्स है, तो वो अभी अभी इस टाइम हम डिस्कस फर्दर कभी करेंगे कि उसमें ओवरडोज का बहुत ज्यादा उसमें नहीं होता, बहुत माइन्यूट क्वांटिटी में इनकी जरूरत पड़ती है, पीपीएम क्वांटिटी में।
ओके सर, थैंक यू।
थैंक यू रितिका। खेतों में he told can they completely replace chemical fertilizer, and one more it was there. No, means completely replace it. It is. I'm not saying that 100% we go for biofertilizer or this PGPM, but selected farmers or selected area which are by default those are practicing, those are not using that chemical fertilizers, they can go for these PGPM or biofertilizers, since uh, that organic products they fetch more price. So not all, but some selected fruits or especially in vegetable crops it can be used, or in leafy greens, these can be used. I'm not recommending, I'm not telling that 100% we should go for these microorganisms and replace all these chemical fertilizers. Not in a single year we can go, but slowly, some amount in 1 year, then next 5 years, then next 10 year, 20 years, some strategic replacement of chemical fertilizer should be there. Like in we are blending that ethanol in petrol. Okay, so we can't say that directly our vehicle it will run on 100% ethanol. Blend it, it should be starting from 5%, 10%, 15%. So 100% ethanol, it will take time. Similarly, the 100% PGPM or that is biofertilizer, it will take time. We can't say that within 1 or 2 years directly replace all these chemical fertilizer with these. Any other query?
सर एक क्वेश्चन था।
Any other query?
सर, अगर हमने किसी प्लांट के अंदर हार्मोन डाल दिया, लेकिन फिर भी अगर वो मतलब कमी उसकी फुलफिल नहीं हो रही, तो क्या करना चाहिए?
Uh, any other query? Is there
Any
some,
other query, participants?
is there any message in text box? Chat.
One
Can
message.
biofertilizer be used with precision technologies? So, why not? It can be used with precision technologies. Not at all a problem. These are as efficient as the other chemical we are using. How can increase the soil organic carbon? Okay, that Manisha also next she told organic carbon, soil organic carbon. These are increasing. One way is that when we are applying organic matter to soil, or these roots or some degraded products of plant are there, so these are decomposed, and some of the volatile carbon it will be lost, but some humus or various very less labile organic carbon it will be there in soil. It will remain for longer years, so soil organic carbon can be arranged. Similarly, some autotrophic microorganisms are also there, like cyanobacteria. So these are fixing atmosphere carbon dioxide, and organic matter is being added to the soil. Some are microorganisms which are forming biofilms. These are secreting exopolysaccharides. So all these are kind of carbon and organic carbon. So soil organic carbon can be enhanced by using these PGPM.
Okay. Any other query, dear participants? And sir, please share us the PPT we will share in the main group.
Okay, I will just I will convert it, and I will share you soon. No problem.
Yes, sir. Now we are concluding our session. Okay. As we come to the end of the today's session, I take this opportunity to express our sincere gratitude to Dr. Vikas Sharma for sharing his valuable knowledge and research insights with us. On behalf of the organizer, EcoFast Agri Solution, and all the participants, I extend our heartfelt thanks to the sir for his time and contribution. Once again, thank you, sir, for your guidance and encouragement. Thank you all for your active participation. We look forward to meeting you again in our upcoming session. Have a wonderful evening and stay safe. Thank you so much, sir, for your.
Thank you. Thank you.
Dear participants, you may leave the session.